Hello, students. Welcome to the renal urinary system, the assessment of the renal urinary system. So we're going to review a couple of terms. The calculi is the stone formation. Continence is going to be the ability to voluntarily control emptying of the bladder or colon. Cystitis, a bladder inflammation, most often with infection. Then elimination is the excretion of waste from the body by the GI tract. External urethral sphincter is going to be a skeletal muscle that surrounds the urethra and helps to control the exit of urine. Basically, this is the anatomy and physiology, right? Incontinence, the involuntary loss of urine or stool. We have the internal urethral sphincter. Is a smooth detrusor muscle of the bladder neck and elastic tissue that helps to control the exit of urine, right? Then we have this term is the microalbuminuria. Microalbuminuria is very important because the albumin is a type of protein that is soluble in water and coagulable by heat. It is found in egg whites, milk, and blood. But the most important about albumin is that it's the magneto to try to hold the fluids inside of the circulatory system. When the person is not getting a lot of albumin, then the fluids are going to go into the interstitial fluid, causing edema. That's very important, and you need to remember that, okay? So then we have the nocturnal polyuria, the renal threshold, which is the point at which the kidney is overwhelmed with glucose and can no longer reabsorb, also called transport maximum. Then we have proteinuria, right? The presence of protein in the urine. And then we have uremia, the buildup of nitrogenous waste products in the blood, which is also known as azotemia, urethral meatus, which is the opening of the endpoint of the urethra. And the urgency, a sense of nearly uncontrollable need to urinate. So let me review azotemia once again. It's a medical condition characterized by elevated levels of nitrogenous waste products such as blood, urea, nitrogen. And this is a concept that I really want you to master. Blood, urea, nitrogen, right? And creatinine, right? So azotemia is a medical condition characterized by elevated levels of nitrogenous waste products such as blood, urea, nitrogen. There you go, the name, and creatinine in the blood. This typically occurs when the kidneys are not functioning properly and are unable to filter these waste products effectively, right? So let's right now move into the anatomy and physiology of the kidneys, and because of that, we are going to play a video, and I love these videos because in a couple of minutes, you can review a huge amount of content from the book, okay? So now we are going to play this video and let's see how it goes. It's 3 minutes, almost 4. Okay, here we go.
Today's topic is the kidney. The two bean-shaped organs located on the left and right sides of the abdomen. They are about 11 centimeters or 4.3 inches long in adults. They receive blood from the paired renal arteries. The blood is then filtered and wastes are removed alongside salt, sugar, and minerals are adjusted. The filtered blood is returned to the body and the waste is converted into urine. Each kidney is connected to a ureter, a tube that transports the urine into the bladder at the kidney's pelvis. Kidney diseases often affect both kidneys. If the kidney's ability to filter blood is hindered, waste and excess fluids build up in the body, resulting in abnormalities which do not give symptoms until late in the course of the disease. These diseases develop slowly over time and cause progressive loss of the kidney's function. Some common kidney diseases are high blood pressure, blood or protein in the urine, diabetes, kidney stone, urinary tract infection, glomerulonephritis, while some are inherited. Anatomy. 3 external layers make up the kidney, and they include the renal capsule, the innermost layer, renal fascicle, the outermost layer. The kidneys are covered by the renal capsule, an outermost covering made up of fibrous connective tissue. This tissue prevents the entrance of infections. 2 layers of fat are attached to the surface of the kidney to help cushion them. They are known as the adipose capsule. The adrenal gland is found sitting on top of each kidney. The right kidney is slightly lower than the left kidney, and the left kidney is more centralized than the right. This is due to the asymmetry within the abdominal cavity caused by the liver. Lower ribs protect the upper part of the kidney. Some important structures connect the kidney to the rest of the body. The renal veins that take blood to the vena cava and the renal artery that supplies blood to the kidney. The renal ileum is the point of entrance and exit of the veins, arteries, and nerves that supply the kidney. The internal layers of the kidney are made up of nephrons. Parts. There are 3 major parts of the kidney. They include the renal cortex, the renal medulla, and the renal pelvis. The kidneys are enclosed by a renal cortex, a layer of tissue covered by the renal capsule and the renal fascia. The cortex provides a space for the hormones from the renal artery and vein to diffuse the nephrons of the kidney. A hormone necessary for the synthesis of new red blood cells is also produced in the renal cortex. The renal medulla. The renal medulla is the inner region of the kidney. The structures inside the renal medulla are known as the renal pyramid. The renal medulla has about 27 to 30 pyramids, with each pyramid containing tens of thousands of nephrons. The renal pelvis. The hilum is contained in the renal pelvis. The hilum serves as an exit and entrance point for nerves and blood vessels. It also serves as a point of exit to the uterus. This layer connects the kidney with the other part of the kidney. Functions. Blood pressure regulation, removal of toxins and drugs, excretion of metabolic waste, regulation of osmotic pressure in the body, balances the body fluids, controls the production of red blood cell, produces vitamin D, which promotes strong and healthy bones.
Excellent.
Thank you for watching our video. Please do not.
Thank you for watching. Excellent. Excellent, excellent. So a couple of concepts that I really want you to remember: the renal cortex, okay, and where is the production of vitamin D. Okay, here you have a very nice graphic about the anatomic location of the kidneys and structure of the urinary system. So first thing, look at the kidneys. They are not in the lower back. They are almost in parallel to the ribs, right? So double check that. Then we have here the bisection of the kidney showing its major structures that we already covered in the video, like the hilum, the renal pelvis, the medulla, okay, and also the microscopic anatomy. Another one that I really want you to remember is the nephron, okay. All of the things that I'm mentioning, you will see them in the exam. Let's watch another video about the nephron, which is the most important, I consider the most important part of the kidney. Okay.
Nephron. Kidneys function as the excretory organ in vertebrates and humans. The kidneys are found in the back of the abdomen cavity and on both sides of the lower back spine. From outside in, it consists of 3 parts: renal cortex, renal medulla, and renal pelvis.
If you see this video, I'm going to go back a little bit. Here on top, you see the lungs. Look where the kidneys are. Okay, it's not at the very bottom of the spine. It's almost in the back of the lungs, in the lower part of the lungs.
Renal medulla and renal pelvis. When microscopically examined, it is seen that kidney is made up of structures called a nephron. Nephrons are the building units of kidney and function as filtering units. Bowman's capsule is found at the beginning part of the nephron. The Bowman's capsule consists of a single layer of epithelial tissue which completely covers the glomerular capillaries. The glomerulus is a network of capillaries which fill the Bowman's capsule. The afferent arterioli entering the Bowman's capsule branch out into the glomeruli capillaries. These capillaries merge together and exit the Bowman's capsule as efferent arterioles. The structure formed by the Bowman's capsule and the glomerulus capillaries are called Malpighian corpuscle. After the Bowman's capsule is the section of the nephron tubule called the proximal tubule. This structure is located in the cortex of kidney. The proximal tubule leads to the loop of Henle in the medulla region of the kidney. The loop of Henle is made of two parts, as the descending limb and the ascending limb. The loop of Henle ascends back into the cortex to form distal tubule. The distal tubule then connects to a wider tube called the collecting duct. While Malpighian corpuscle and proximal tube parts of the nephron are found in the cortex, the loop of Henle is located in the medulla region.
Thank you so much. So it is very important now, since we are entering the graduating level, okay, that you master your map. Your map, I always tell you, you need to know your map. There's no, I don't think it's safe to be graduating without knowing your anatomy and physiology, right? So we spoke about the nephron, and the purpose of this nephron is to concentrate urine during times of low fluid intake to allow continuous excretion of body waste with less fluid loss. Extremely important concept, right? So the nephrons are going to create the balance that is needed. Now, it's important to know where the Bowman capsule is located, right? And what is the definition? It's a sac-like structure that surrounds the glomerulus. Okay, the urine in the collecting ducts passes through the papillae and empties into the renal pelvis. So it's important to remember the renal concepts again, the nephrons, the glomerular filtration rate, concepts like the glomerulus, right? Those are the most important. Renin is another huge concept that you need to master. Renin converts renin substrate, which is the angiotensinogen, into angiotensin 1, right? This leads to a series of reactions that cause secretion of the hormones aldosterone, right? So now I want to review the urine formation with you guys, because this is important as well. Okay, here, let's play this video. Urine formation. So we review the anatomy of the kidneys, the nephrons. Now we're going to review the urine formation. Pay attention, because this is, for me, the most important part of this entire chapter.
There are three processes that are involved in the formation of urine. The first process is glomerular filtration, which is the separation of smaller substances in the plasma, such as glucose, amino acids, ions, and water, from larger substances in the plasma, such as platelets, white blood cells, red blood cells, and proteins. The second process is tubular reabsorption. Tubular reabsorption is the movement of substances from the tubules of the nephron into the peritubular capillaries. And finally, the last process of urine formation is tubular secretion. Tubular secretion is the movement of substances from the peritubular capillaries into the tubules of the nephron. Glomerular filtration occurs at the renal corpuscle. Here, substances are delivered to the nephron via the afferent arteriole and into the glomerulus. Smaller substances easily diffuse across the membrane of Bowman's capsule. leaving behind larger substances in the glomerulus, which will eventually be moved into the efferent arteriole. The smaller substances in Bowman's capsule is now called the filtrate, and will move through the kidney tubules. Depending on the body's needs, these substances can be reabsorbed or added to be eliminated as waste. After Bowman's capsule, the filtrate moves into the proximal convoluted tubule. Taking a closer look at the proximal convoluted tubule, we see that substances such as water, glucose, amino acids, ions such as sodium and chloride are reabsorbed back into the body via the peritubular capillaries. From the proximal convoluted tubule, substances then move into this U-shaped structure called the loop of Henle. And there are two parts to the loop of Henle. There's the descending limb and the ascending limb. Taking a closer look at the descending limb of the loop of Henle, we notice that there is further tubular reabsorption of water. And in the ascending limb of the loop of Henle, we see further tubular reabsorption of sodium and chloride ions. After the loop of Henle, the remaining substances enter the distal convoluted tubule. In the distal convoluted tubule, we see the tubular secretion of ammonia, hydrogen ions, potassium ions, and drugs. And finally, after the distal convoluted tubule, the substance is now called urine enters the collecting duct. And at the collecting duct, we see the last of tubular reabsorption of water. And this is where the dilution or the concentration of urine occurs. Once urine has been produced by the kidney and transported to the urinary bladder, it's now time for urine to be expelled from the body. Micturition is another term used for urination. The urinary bladder is composed of transitional epithelial tissue and smooth muscle, and embedded in the wall of the urinary bladder are sensory receptors or stretch receptors that are attached to these sensory neurons that'll synapse at the spinal cord with motor neurons that will stimulate the contraction of the smooth muscles that line the bladder walls. When the bladder becomes distended because it is filling with urine, the stretch receptors within the walls of the bladder send an action potential down the sensory neuron, and that action potential will carry on to the motor neuron, causing the smooth muscles of the urinary bladder to contract. Another action potential from the sensory neuron will be sent up the spinal cord to the motor cortex of the cerebrum. From there, another action potential with motor input will be sent to another motor neuron to stimulate the opening of the urinary sphincters. And there are two urinary sphincters. There's an internal sphincter, which is the sphincter closest to the urinary bladder. It's composed of smooth muscle and under involuntary control. And an external sphincter, which is composed of skeletal muscle, which is under voluntary control. So as the smooth muscles that line the urinary bladder contract, the sphincters, the urinary sphincters, relax and open, allowing urine to pass through into the urethra.
Okay, very interesting concepts, because right now when we are going to talk about incontinence, we know that there is a significant role from the nerves, right, to control the involuntary and the voluntary sphincters for this. Okay, so renin, I want to be precise on this, but I think I'm going to start from behind. Special cells in the afferent arteriole, right? Efferent arteriole and distal conduct tubules are known as juxtaglomerular complex. So that's the figure 60.4, right? 60.4 is here. And we are going to see the efferent arteriole, you see, is the one that is heading to the glomerulus. And this is the afferent, and the efferent is the one going out from the glomerulus, right? So now that we have clarified this, let's go back to the theory, right? These cells produce renin, which is a hormone that helps regulate the blood flow, glomerular filtration rate, and blood pressure. So again, the special cells in the afferent arteriole, and the distal collecting tubules are known as the juxtaglomerular complex, right? And they are going to produce renin, which is a hormone that helps regulate blood flow, glomerular filtration, and blood pressure. So far, so good? Okay, so now renin is secreted when sensing cells of the DCT, right, called the macula densa, the distal collecting duct, sense changes in blood volume and pressure. Again, so renin is going to be secreted when the renin is sensing changes in the blood volume and pressure. Okay, when there is hypotension or less hypovolemia, renin is going to be reacting, right? The macula densa touches the renin producing cells. Now, renin is produced when the macula densa cells sense the blood volume, blood pressure, and blood sodium level is low. Okay, we know that. So the most important is that renin then converts renin substrates, right, into angiotensin 1. So renin, when renin is sensing a decrease in volume, or blood pressure, is going to convert into angiotensin 1. That's the shortcut, right? Forget about the renin substrate. It's going to convert into angiotensin 1. This leads to a series of reactions that cause secretion of the hormone aldosterone, right? Aldosterone increases kidney reabsorption of sodium and water, because by reabsorbing sodium and water, the blood pressure and the circulation is going to increase, restoring blood pressure, blood volume, and blood sodium levels. Okay, it also promotes the excretion of potassium. So renin is going, if we are increasing sodium, we are decreasing potassium, because there is a inverse relationship between sodium and potassium. Okay, so that's important. Renin at the end is going to help regular blood volume, blood pressure, right, and the reabsorption of sodium. Okay, so now the role of aldosterone, renin, angiotensin 1, and angiotensin 2 in the renal regulation of water and sodium. So this is good to know, the renin, angiotensin, aldosterone system, okay? And I am going to play this one because you cannot graduate if you really don't understand the renin, angiotensin, aldosterone, okay? It's very important. Okay, here we go.
The renin-angiotensin-aldosterone system, RAS, is a hormonal system that controls blood pressure. While baroreflex is a short-term response to sudden changes in blood pressure, RAS is responsible for long-term regulation. In the kidneys, within the walls of afferent arterioles, there are specialized cells producing prorenin called juxtaglomerular cells. Upon activation by a drop in blood pressure, prorenin is cleaved to form renin, which is released into the blood. Renin converts a plasma protein called angiotensinogen, produced by the liver, into angiotensin 1, a peptide of 10 amino acids. Angiotensin 1 is further converted into angiotensin 2, an 8 amino acid peptide, by the angiotensin converting enzyme, ACE, predominantly present in the lungs and kidneys. Angiotensin 2 is a hormone. It binds to angiotensin 2 receptors in tissues to exert various effects. It stimulates vasoconstriction in systemic arterioles. It promotes sodium reabsorption in proximal convoluted tubules of the kidneys. It induces the release of aldosterone from the adrenal cortex. Aldosterone promotes sodium and water retention in the kidneys. In the central nervous system, Angiotensin II has several effects. It acts on the hypothalamus to stimulate thirst and encourage water intake. It induces the posterior pituitary to release antidiuretic hormone, which promotes water retention by the kidneys. It reduces the sensitivity of baroreceptor response to increased blood pressure, so that this response would not counteract the effect of RAS. All these actions lead to an increase in blood volume and blood pressure. Angiotensin II is short-lived with a half-life of 1 to 2 minutes. It is degraded into angiotensin III and IV, which have lesser effects. Overactive or inappropriately activated RAS is a cause for hypertension. RAS is a frequent target of antihypertensive drugs. ACE inhibitors and angiotensin receptor blockers are common treatment for hypertension.
Thank you so much. The most important is that that's why I'm going to leave this video here so you can review it. Okay? So you cannot work in ER if you don't understand the RAS. You know, your renin is going to be activating angiotensin 1, which is going to hold, okay, the sodium, and is going to increase water reabsorption in order to bring the hypovolemic hypotension state into normal blood pressure. And angiotensin 2 also is going to participate there. Now, that's why ACE inhibitors, right? Inhibitors are going to stop that reabsorption. And those medications are used to control hypertension, which is the excessive amount of fluid and blood volume, right? And blood pressure. So you need to understand this RAS. Many other organs are participating, like the liver and the hypothalamus. So please review this video, okay, in order to understand that, because it's NCLEX alert. Now we have the function. The kidneys have both regulatory and hormonal functions. We know that, right? The regulatory functions control fluid and electrolyte balance and acid-base balance. The hormonal functions control red blood cell formation, blood pressure, and vitamin D activations. Again, the hormonal functions of the liver controls red blood cell formation, that is your erythropoietin, blood pressure, and vitamin D activation. Okay, so now let's review your acid-base balance video. Also, it's very important for you to understand. Okay, I find very interesting to play video for this. You're gonna love this video.
Hey everyone, I'm Kendall Wyatt, the content director here at Big Money. Today we're going to do a high-level, brief synopsis of acid-base imbalances. I know everyone's favorite subject, mine too, of course. Now, we're going to go through the lab values, and then we're quickly going to go over how you can briefly guesstimate an acid-base problem. Now, when you feel like you're solid on the content, head over to our Mastery Challenge, where you can really see if you remember all of the facts for long-term retention. And of course, it's free. If you don't feel solid on the content yet, you can always visit one of our webinars, where we go into much more detail, explaining more crazy things about all the nitty-gritty details. After that, be sure to download our absolutely free outline and study guide covering acid-base imbalances. Now, let's get to those details. Now, the lab values we're going to talk about are pH, CO2, and bicarb, or HCO3. The normal pH of blood is 7.35 to 7.45. That's the normal range. Now, of course, below that is acid, and above that is alkaline. And you look at pH, and you have to assess whether it's up or whether it's down. Well, if it's up, then that's alkalosis, or the alk loser inside of pykmonic. If pH is down, or less than 7.35, you know that's acidosis, or our acidic limit inside of pykmonic. CO2, what is a normal CO2? Well, pH is 7.35 to 7.45. Okay, I got that. Memorized one. Oh, I gotta memorize all these others. Well, you can easily remember acid-base because pH is 7.35 to 7.45. So CO2 is 35 to 45. Those are pretty much the same numbers. Remembering 35 to 45, that's what you need to remember. CO2 is an acid. Now, how do we remove and regulate CO2 in our body? Well, we blow it off. That's blowing off some acid. So 22 to 26 is bicarb. Now, bicarb, remember bicarb is regulated by the kidneys, and it's slow to react. So remember, you get angry and you huff and you puff and you blow? Well, you can do that really fast. But it takes a long time, sometimes 24 hours, for your kidneys to be able to begin compensating for all your angry and huffy puffiness. Now, let's talk about an actual acid-base problem. I want you to look at that pH and I want you to say, is it within 7.35 to 7.45? If the lab value is below 7.35, well, that's an acid. If the number's above 7.45, that's basic or alkaline. So I want you to look at a problem and assess the pH. If it's below 7.35 and trending downward, you need to put acidosis. If it's above 7.45, write down alkalosis. And put that down in your problem right away, so you already got 50% of the problem. You've got half the answer right there. Step number 2 is to look at the CO2. The CO2 level. Now, remember what regulates the CO2? Breathing, right? Remember, if you get mad and you get upset, you're going to blow off a lot of CO2. Remember that. So when you look at the CO2, I want you to look at it and say, is it between that normal range of 35 to 45? And if you say no, well, it's abnormal, right? And look at pH. Assess the two. If CO2 is going the opposite direction of pH, one up and one down, or one down and one up, they're going in opposite directions. Why is this important? Well, this makes it super easy. If it is, that's a respiratory problem. Well, if we already have acidosis or alkalosis written down, then you already have your answer. It's a respiratory alkalosis or a respiratory acidosis. You have one of the two answers right there, and for the most part, with all the basics, you're done. Now, what if they're both going in the same direction? Then we need to look at bicarb, HCO3, and only do we look at it then. Because you can remember that metabolic problems, metabolic acidosis, and metabolic alkalosis are all down, 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 or up, up, up. It's really kind of that simple. Now, it does get more detailed when we talk about compensation. But right there is going to be able to get you 80% of the problems right every single time. Now, for compensation and all the other details, you can add those on later, and once you understand this concept, you can actually add them on pretty easy. For all the other details and everything you need to know, even a wonderful Picmonic with all kinds of graphical details, you can play it over and over. You can even go and have it as a bedtime story if you like. And those are all the details and the fine little things that you can put together in our study guide with our Picmonics, or by joining one of our webinars if you can catch one that we have scheduled pretty regularly. You can check us out at picmonic.com. Go and sign up. It's a free trial.
Thank you so much. So that was like a brief review about metabolic acidosis or alkalosis, or respiratory alkalosis or acidosis, right? It's everything about the level of the pH, the level of the CO2, or if it's bicarb, right? And we know the CO2 is regulated by the lungs and the bicarb is regulated by the kidneys, okay? Try to review this video because next semester is going to be a lot in reference to this, okay? You're going to be tested on that. Regulatory functions, the kidney process that main fluid and electrolyte balance, we review that. Glomerular filtration is the process in urine formation. You need to remember that, right? And also water, electrolytes, and other small particles like creatinine, urea, glucose are filtered across the glomerular membrane into the Bowman capsule to form glomerular filtrates. As the filtrate enters the proximal convoluted tube, it's called tubular filtrate or early urine. Large particles such as blood cells, albumin, and other proteins are too large to filter through the glomerular capillary walls. Therefore, these substances are not normally present in the screening final urine. Remember your RBC formation, the outer layer of the kidneys where the nephrons blood filtering units begin, right? The renal cortex, remember that definition, also creates the hormone erythropoietin, which helps make red blood cells in your bone marrow. Erythropoietin, renal cortex. Here we have the diagram of the glomerular capillary wall. Okay, so you have the Bowman capsule. And then glomerular filtration rate is controlled by blood pressure and blood flow. And the kidneys self-regulate their own blood pressure and blood flow, which keeps glomerular filtration rate constant. So let's double check this table. 60.1 vascular and tubular components of the nephron. Most important that I want you to remember really are the glomerulus, okay? The glomerulus, okay? Site of glomerular filtration. Glomerular filtration occurs when hydrostatic pressure, like the blood pressure, is greater than opposing forces, tubular filtrate and oncotic pressure. Also, I want you to remember the loop of Henle, okay? Is important, is the regulation of water balance. So we don't need to know all these in particular, but for example, this is the vascular component, okay? The glomerulus is the vascular components, right? And then you have the tubular components, and from that tubular components, I want you to remember the bone capsule. This one is very important, okay? And also I want you to remember the loop of Henle, okay? Loop of Henle. The way I remember the loop of Henle is L is low, H is high. So it's a structure that has a low tube, a tube that is going to the lowest part, and high, H is high, so it's going low and high, okay? So that's how I remember the loop of Henle, right? Also, what else? Let me see here. Okay, let me see what else I want you to remember. I think it's enough in terms of the tubular components, right? So the tubular components. And then, so let's stop here. Let me see if we can move this farther. So glomerular filtration rate, glomerulus. The Bowman capsule collects glomerular filtration rates and funnels into the tubule. That's why I want you to remember, because they are interconnected. And then the loop of Henle, which participates on the regulation of water balance. That's extremely important, okay? So review the videos, and when we come back, we are going to continue with the remaining part of this chapter. Thank you.